Ka tangi te titi, ka tangi te kāka, ka tangi hoki a hao te he Māori ora. Ina mana, ina reo, aura rakatira mā tēnā koutou, tēnā koutou, tēnā koutou katoa. Tēnā te raru e te maru o ka mana whenua, ka te māmoe waitaha kaitahu no mai hara mai tauti mai. I stand here under the umbrella of the people of this place, ka te māmoe waitaha and kaitahu, to welcome you here. Ki te whare tūnei, tēnā koe, tūtonu, to the house that stands here, greetings to you. Ina mate haere hoki atu mā ki te pō. To those loved ones that we may have lost as a community or for any of you individually, may we remember you and may you go in peace. Ki te rangatira eharaki, to the professors in our official party, tēnā koutou, Professor Hook, Bremer, Gemmell and Monk, tēnā koutou, and to our special honoured guest, Professor Tyndall, tēnā koe. Ki te manuhiri i te whare wānanga o tākou, ki te manuhiri te Otopoti, te Te Wai Pounamu, te Hōwei Whā, no mai hara mai welcome to those visitors here to the University of Otago this evening, to those from Dunedin, to those that have come from other parts in the South Island, to those from the Four Winds, my great welcome and greetings to you all. And in particular, we have some very special guests in the audience supporting Professor Tyndall, and online. We have your mum, Geraldine, Father Alan and his wife Di here. Online. You have your brother Dean and his wife Kelly visiting from Melbourne, so welcome to the warmth. This is probably like a normal Melbourne day for you, right? So I get that. To Trudy, Lara and Sam also welcome from the university. Ko waia, who am I? No Otopoti hao, ko Katarani te iwi, ko Richard Blakey, tāko ingoa he kaimahi ao te whare wangana o tāko. So I'm Professor Richard Blakey. I'm Deputy Vice-Chancellor for Research and Enterprise, and because our Vice-Chancellor, Professor Helen Nicholson, is away and gives her apologies, I'm actually acting Vice-Chancellor at the moment, so I can be here officially to welcome you on behalf of the university. And just to give you a little bit of an overview of what we're here to celebrate through an inaugural professorial lecture. This is one of the occasions that we do get our gowns on and we do love as academic leaders in the university, because it gives us an opportunity to celebrate promotion or appointment to the highest academic rank at our university, that of professor. This is an aspirational rank that requires exceptional performance and leadership in all three domains of the role of an academic: in research, in teaching and in service. And you'll hear from Professor Hook when she introduces Joel the kinds of things that he has done to get that service. I won't steal her thunder, but just a couple of remarks that what makes your achievements very remarkable, Joel, is how you do balance those commitments. You are an excellent researcher who we are very grateful that you've brought significant amounts of research funding into the university from a number of sources, from Blue Skies funds, from the Health Research Council and NIH and MRC in Australia, but also from industry. So your research is there to be applied. You're an outstanding and very popular supervisor, having 21 PhDs under your supervision or jointly supervised with other. You're an excellent teacher. I note particularly that you've taught into 3 of our divisional programs, into health sciences, into chemistry programs, and even guest lectures in drugs and the law, I think it was. And what I want to give particular note for, what I want to particularly note is your community service. For us as an academic community, often that is service on editorial boards and committees and other things, and you do that at the highest level. But I also know that you serve your family and the communities of Dunedin very, very well through surf lifesaving and other things, and it's a real hallmark of a career well spent that you have balanced those commitments to the things that are important to you, both inside and outside academia. So I'd like to ask Professor Hook to come and give you a formal introduction before you give your IPL, but please accept on behalf of the university our wholehearted congratulations for your well-deserved promotion. Thank you very much.
Tēnā rā, tēnā koutou. Hei mihi nui, hei mihi mahana kia koutou. Ko Sarah Hook toka ingoa. So my name's Professor Sarah Hook and I'm a colleague of Professor Tyndall's at the School of Pharmacy, and it's my great pleasure and honour to provide this introduction for Joel's inaugural professorial lecture. And I've known Joel for many years now, since he started at the university some 19 years ago. Joel is a medicinal chemist, as I'm sure you all know, with expertise in drug discovery using computational methods. He is Australian, but we have almost forgiven him for that, since he has married a Kiwi and both his children are Kiwis. Joel grew up in Melbourne and has a BSc Honours in Chemistry from Monash University. He then moved to the University of Queensland's Centre for Drug Design and Development for his PhD, which is also where he met his not at that stage, but his soon-to-be wife, Trudy. His time in Queensland was obviously very successful from both a personal and professional standpoint. And one of his scientific works, which he's most proud of, was based on work carried out at that time. It was based on work looking at understanding Alzheimer's disease. And this was during his PhD, and it was published in the prestigious Journal of Biological Chemistry. It's been cited over 980 times and is still cited today. In 2000, after finishing his PhD, Joel moved to work at the Institute of Cell and Molecular Biology at the University of Edinburgh. He enjoyed life in the vibrant little city and also managed to get 2 publications while making most of the nightlife. It also possibly prepared him for the change in temperature he could expect when he came to Dunedin. However, he could not stay away from the University of Brisbane and, more importantly, Trudy, who had remained there to complete her PhD. And he returned to Brisbane in 2001 to take up a position as an independent researcher. It was also during this time that Joel and Trudy married. They also carried out research together. Some of their research was based on potent neurotoxins produced by snails, which happen to live in the waters around the Great Barrier Reef. And as part of this research, they went on a week-long snorkelling trip to collect samples. Doesn't sound like a work to me. And it wouldn't have been for Joel, who has a love of swimming and the water. This has survived as moved from the balmy waters of Australia to the frigid waters of Dunedin and the Southern Lakes. But apparently he does now require the use of wetsuits. As Professor Blakey mentioned, Joel is a stalwart of the St Clair Life Saving Club and has passed his love of water on to his daughter Lara, while his son Sam has inherited his skill with computers. In 2004, Joel took up a position as lecturer in medicinal chemistry here at Otago in the School of Pharmacy. His research here involves understanding proteins as potential drug targets. A key feature of Joel's work is that he is highly collaborative and is an integral part of teams looking at this area. And it does sort of show Joel's social and people-focused nature, that he has been able to maintain these strong collaborations and teams with groups within and outside the university for long periods of time. So Professor Tyndall has been an integral part of multidisciplinary teams investigating the biological basis of disease and developing new drugs, which I'm sure he would tell us about tonight. This research has resulted in Joel being a named investigator in grants totaling more than $16 million and more than 100 publications in international peer-reviewed journals. He has gained an international reputation in his field of computational modelling, which has resulted in being recently asked to write an expert review on a new COVID drug in the prestigious Journal of Medicinal Chemistry. Another highlight of his research career so far is his contribution to a team identifying a mechanism of resistance in a class of antifungal drugs. Joel's collaborative approach to life also extends to his leisure activities. He has also, for the last 10 years, competed in triathlons, but sensibly does not do the whole thing himself. Instead, he completes a swimming leg, which is his area of expertise, and lets his teammates Dave and Danny do the rest. He has also recently taken up wakaama paddling. However, Joel's sporting endeavours did take a pause last year when, after one too many last runs down the ski field, he ended up in Dunedin Hospital with a broken tibia and fibia. Time does catch up with us all, Joel. Joel is a dedicated teacher and he has earned the respect of both undergraduate and postgraduate students. His PhD students have gone on to positions in industry and academia, with the recent graduate Jasmine Wang recently becoming a Chief Scientific Officer in a biotech startup, based on the start that Joel gave her. Joel has close industry links and has carried out contract research and acted as a consultant for a number of Australasian companies. The third key role of an academic is service, and it's perhaps the one we don't talk about enough, but it's where Joel has shown particularly strong and sustained leadership. Firstly, taking on the role of Associate Dean of Marketing, Communication and Engagement at the School of Pharmacy, and then taking on the role of Associate Dean International for both the School of Pharmacy and the Division of Health Sciences. This is a key role for the school and the university, promoting our courses and our research internationally. This role requires someone with excellent communication skills and interpersonal skills and a great deal of patience. It does also at times require a lot of travelling, and we've been very pleased that Joel's family has put up with the time he's had to spend away from home. Joel has strengthened relationships with universities with strong health science programs in the Asia Pacific area, and the university, and in particular the School of Pharmacy, has benefited hugely from linkages he has made. Right, so I think that's now time for me to finish and move on to the main event. I am personally very excited to hear about Joel's achievements in the area, and I would like to welcome Professor Tyndall to the podium to talk to us about looking at molecules and what they do in disease. Welcome, Joel.
Thank you very much, Sarah, for that introduction. I can tell you now it feels a bit different being in the spotlight rather than just coming to watch. People say enjoy this, so let's have some fun. So, my name is Joel Tyndall. As you can see, I did originally come from the lesser known West Island, as you can see up there, from a small place called Melbourne, over there. I grew up in Melbourne, trying to identify photos as people I want to put up in these types of lectures of my primary school was quite difficult because they're all quite modern. So the nearest thing to that was the Lobster Cave, which was a 70s icon growing up in the 70s around there. There's a few in the audience who'll understand the amusement there. So growing up just down the road from the Lobster Cave, going to primary school pretty much behind the Lobster Cave in Beaumaris Primary School. Didn't have to go far to go to secondary school, went to Mentone Grammar. There, a boys' school, which rightly or wrongly set me on the path that I have ended up here. Now it's a co-ed school. Whether or not I was able to talk to girls at that stage wouldn't have mattered either way. I didn't even go far for university. I turned around and decided that, well, we'll go to Monash University in Clayton. There's nothing wrong with that. It's a very good university and set me off well. As has already been mentioned, I like water. This was where I grew up. This is at Ricketts Point. So growing up, we had the luxury of watching the sunset go down over the water. And I'll get back to that as we go. So I embarked on my university career in a Bachelor of Economics. That seemed something that would challenge me at the time. Work out which ones are my notes. A little known fact about me in fifth form, we only had 6 forms in Melbourne. In fifth form, I did law and managed to do very well in that paper, only to drop it in my sixth form. But I thought that was possibly a good opportunity to go there. Life told me otherwise, and after 2 years of doing economics, it was decided not to continue with that, or shall we say, someone else decided that I shouldn't continue with that. We managed to get back into applied science at a lesser known institution, Caulfield Institute of Technology. It was a very relatively prestigious university in its, institute in its day, but as you can see down here, the architecture, or the more modern architecture, wasn't as attractive as can be. I do remember it does have some appropriately attractive architecture from around the 40s and 30s to 50s. I won't look at my brother to confirm which year it was. I don't actually remember ever going into that building, however. Ironically, having Monash asked me to think about my career in a different manner, in 1990, I believe, Monash took over Caulfield Institute of Technology. So I was back at this institution. I did start in the Robert Menzies building, with economics, but I moved to the smaller science wing to start my Bachelor of Science, or continue my Bachelor of Science. I guess ironically, given that it's medicinal chemistry, which has a lot of biology involved with the science, I managed to get a conceited pass in first year biology, whereby I'd passed, but they wouldn't let me do it again. I also decided that I probably wouldn't need computer science either, ending up as a computational chemist. Nonetheless, in second year, I went on to do some chemistry and some mathematics papers, and pushed on into third year into chemistry, realizing that as I put it to myself and to others when they asked, I continued with chemistry because it didn't annoy me, and then it realized that I could actually do it. So I completed my Bachelor of Science, and in third year, this is probably the catalyst. One of the professors there, Patrick Perlmutter, who was Professor in Organic Chemistry, had an assignment associated with these silicon graphics computers, which around 93, 94 were worth to purchase, I think, about $10,000 US. And what we were tasked to do was to simply visualize the protein and ligand complex, something similar to what's on the far left, and identify exactly what the interactions were. So the ligand-protein interactions. And these computers also were used in the development of the Terminator 2 movie. So they were pretty flash, pretty expensive. And I do have a prop. Steve, it's off. But it was off. You can pretty much do the visualization of those proteins on a phone now. But these were fairly impressive pieces of tech. So I completed my Bachelor of Science at the University of, Monash University. As students are wont to do, and what is still available now, which I think is a great opportunity, is summer studentships. So having finished my bachelor's degree, I went for a summer studentship with ICI, which I had to remember first of all what the acronym was, and then I had to look up what that meant. And it's not on my notes. Imperial Chemical Industries, I believe, which was then taken over by Orica. And that was effectively making brown detergents not brown. So that didn't quite excite me, so I continued on with my honours degree. We didn't have the opportunity to look at any molecular visualization or protein stuff, at least in the chemistry department, as an honours project. So this was probably my first introduction to medicinal chemistry or biological type chemistry, chemistry associated with drugs, whereby the title, and I wondered why I carried this around for most of my life, but I think I've just worked it out. I can probably get rid of it now. That's my honours thesis, and it's entitled Synthesis and Anti-Cancer Activity of Platinum-2 Organoamides. So that is the technical term for derivatives of cisplatin anti-cancer drugs. And this was with Professor Glenn Deacon here, and this is a photo taken from somewhere in the web, when the lab was delivered one of the first dry glove boxes. Now this box is used for chemistry which is oxygen sensitive, so they can put an argon or nitrogen environment, and I believe it took 15 minutes to purge, possibly each time at that stage. And there was a number of those by the time I got into the lab at that stage. Professor Deacon was an expert on lanthanide chemistry, and if people are able to picture the periodic table, which I know fairly well, the list down the bottom is lanthanide chemistry. Glenn Deacon was an expert in lanthanide chemistry. I avoided that, thankfully, and went into some platinum stuff. At the end of honours, based on Patrick's suggestion, I went up to Queensland to the Centre for Drug Design and Development on a summer studentship, whereby I sat and mused with a lot of visualization of molecules, in particular looking at anti-cancer drugs and how they interact with DNA, and that's their known mechanism of action. I also made some peptides. That's about all I can remember doing, apart from probably going to see a few bands and probably partying in a few other areas too. And I came back to Monash to start my PhD. But it wasn't long after that I realised that doing a PhD in platinum anti-cancer drugs wasn't quite what I wanted to do. So I actually stopped my PhD at Monash around the same time that one of the PIs up in Brisbane, David Fairley, rang me up and said, why don't you come up here? So I moved up to University of Queensland to start a PhD up there. And whilst it says 18 hours, I think it took me a week because I stopped off to see some colleagues on the way also. And also as I was, one goes through a lot of self-reflection in these times, preparing these lectures. And looking at that, there wasn't much internet or maps. So I'm trying to work out how I actually got from Melbourne to Brisbane to the actual specific places that I needed to go without Google Maps and not having a large book of other maps. So that was my journey to start my PhD, or start again my PhD. And this is where I continued. So there was involved some synthesis, and we'd set out to make the smallest cyclic peptide around. And what I'd found trying to convince my supervisor that I couldn't purify this gel because I'd made this tetrapeptide, 4 amino acids. It was hard. It's like, well, can you try something else? Little did we realize that peptides forming gels could be useful in other areas. So that was probably the last time I was in the lab doing synthesis. I then went on to do some other investigations, particularly around Alzheimer's disease. Looking at how metal ions interact with the specific peptide, which is shown over here, beta amyloid peptide, because it's understood to create reactive oxygen species. I did some overnight experiments, numerous late nights at a centre for magnetic resonance, I think it was called, using EPR, which I've actually just forgotten what it means. And I don't really understand what that means, but we've managed to work out some things with Alzheimer's disease. Needless to say, that's still a big challenge these days. Part of my research also was to look at a large class of enzymes called proteases. Now, these proteases are the enzymes that chop up other proteins. They're 4% of the human genome, and it's the largest enzyme family in vertebrates. And we know that they exist in other organisms as well, not just vertebrates. So I did a large analysis of what binds to proteases. And basically what I'm going to talk about is a lot of my research, which basically is broken down to squiggles and lines. Easy to simplify it. Specifically, we did look at some structural aspects of antiretroviral drug leads, looking at HIV protease, as well as molecules interacting with target proteins associated with inflammation. So some of the finer details. So this was the paper that Professor Hook alluded to, copper potentiation of Alzheimer's Alzheimer beta amyloid neurotoxicity. I am very proud of that. Significant paper. It is literally an Australian football team on the author list. I'm 5th out of 20. That includes the reserves. So I then moved away from the Alzheimer's peptide, still retaining the knowledge for some of the teaching that I've done in the past 20 years. It sounds like a long time. Looking at these proteases, these proteolytic enzymes, and trying to work out if there is something similar that they all do, that we can use that knowledge with respect to developing drugs that may target these types of enzymes. And as I said, here is the line with the squiggle and the line of what we've been looking at. Specifically, HIV protease was one of the key things that we looked at. As an aside, I try and mess with people's heads by having these things spinning, and hopefully they may feel a bit weird, but now that I stand up here and look at that, I'm feeling a bit weird. So that's a relatively simplified and sort of glorified pretty picture of what we were doing. So HIV protease is was well known at the time. It was at the forefront of disease and drugs also. But we were able to look at understanding how the general class of proteins recognise those ligands, and also how we can constrain into that motif, so the line, such that it retains that. It was around the same time that a flatmate of mine, who was also doing his PhD, Michael Kelso, suggested that we go swimming in the off-season being winter, because he was a surfer and he needed to keep fit for surfing. And being able to swim in an Olympic-sized pool in the open is a fantastic experience, even if it is only slightly heated in winter. We'll come back to that as we go further. So I finished my PhD and managed to organise a fellowship and a postdoc in Edinburgh. There were a number of other opportunities there, but Edinburgh was where I decided that I'd go. And as Sarah mentioned, Trudy remained in Brisbane at the time to finish her PhD. So it wasn't a rush postdoc, but it was a short one. So as a scientist, I guess we are recognised by signposts such as publications. So yeah, there was 2 publications that came out of my time there. But there was a lot more science that I got involved in. So the supervisor up there, Malcolm Walkinshaw, used to work in a pharmaceutical company, Sandoz, in Europe. He also wrote his own software as well. So it was great to learn new aspects and new theories from him around that type of technology, and also the developing technology of high throughput screening using computational methods. Rather than going and purchasing large amounts of compounds to then screen physically in the lab, you can do this computationally, such that you don't have to buy 10,000, you can maybe buy 100 of those, with some more certainty that you might get something that's going to work. So whilst we have the signposts of our publications, I gained a lot more out of Edinburgh. And a lot of people do also put pictures of themselves and things like that. I don't have that many, or either that or I chose not to go and get them. But at least I did find, my flat was probably about here, on Marchmont Road in Edinburgh, and you could actually look up out of one of the windows. It was the box room, which are the smallest rooms in Britain. You could fit a single bed and not much else in there. If you looked out that window, you could actually see Edinburgh Castle, which was pretty cool. So I was coming towards the end of my postdoc and not quite really getting the whole idea of applying for jobs. I emailed my PhD supervisor and said, "Can I have one?" And thankfully he said, "Sure." So I came all the way back to Brisbane. As Google said, it takes a day. Probably took a bit longer than that. I do remember catching up with a very good friend in Prague for a week before we came home. So this was David Fairley, my PhD supervisor, who helped me a lot. And these 2 papers were helping an honours student that I was given co-supervision of at the time. Again, we're looking at the proteases, so these proteins exist through us, and they're quite common in viruses as well, as we've mentioned. So these 2 papers here talk about West Nile virus, which is still a problem, along with dengue fever, in the tropics in particular. So at that time, crystal structures, so by the nature of that, you take a molecule, in particular a protein, crystallize it, and that gives it order such that you can then determine what the protein looks like. There wasn't, is it 100,000? That's one run with 100. There wasn't 100,000 crystal structures available as there is today. But there was a select few from a number of different species or compartments that were very similar to other things. So I can say that dengue fever, or the virus associated with dengue fever, is a very similar virus to West Nile. They're in the same class. So we were able to build models of these proteins based on similar proteins that we know the structure of. We're able to actually dock in the substrate such that we can then understand how the protein works, to take it to the next step to develop molecules that are going to block the processing or the action of this enzyme in the disease state. So the context of homology, of homologous meaning the same, we've got a common structure within classes. I was also beginning to understand that genetic makeup of viruses is similar. Was I understanding then? I'm not actually sure. I wrote this week, obviously. So now I understand that viruses are quite similar. So this was, I guess, my second postdoc back in Brisbane, and this was a very important collaboration that had developed over time with this very good scientist here, Dr. Trudy Milne, whereby we were looking at, Trudy was looking at cone snails, and these things look friendly and look pleasant, but they hunt. They don't hunt people. They hunt fish, they hunt snails, and you can get very sick, and people can die from the venom from these snails. And I was very lucky to go to, I've forgotten the name of the island, but we'll get to that a bit later, but it's just off Gladstone in the Great Barrier Reef. I was taken on that cone snail collecting trip as catering officer. We did actually have a film crew who wanted to do a documentary also, so that was a bit of fun, and they took us out to some of the outer reefs, all for scientific purposes, of course. But nonetheless, so this is all Trudy's work, but I was involved in generating models of trying to understand what these enzymes did. This particular protein exists in a lot of different venoms. Again, homologous stuff. But this type, there is also a class of proteins, the human crisps, which is associated with fertility and human sperm from memory. So lots of similarities. So I maintained that collaboration and also that love of water. So these two papers I have always had at the forefront of my mind with respect to what I look at and what I go back to and what I do. So for want of a better term, like the journal that they came out in was Chemical Reviews. It wasn't simply a review, it was more of a meta-analysis. So I did this as part of my PhD and continued on, but looking at these proteases, at that time I looked at every single crystal structure of proteases to identify what they recognised and how they recognised. And they recognised molecules in this line format. We also looked at another very separate class of proteins, G protein-coupled receptors. So I mentioned that proteases make up about 4% of the human genome. I think these receptors make up about the same, but significantly they make up about 30 to 35% of all drugs available on the market. Things like antihistamines, asthma drugs, salbutamol. I won't go into that. There is lots of them, but they're very common. So taking that forward, moving into some of the extra work that I was doing, it's been mentioned that I collaborate. So my work is very collaborative. So we looked at cyclic peptides. Specifically, this was looking at cyclic peptides forming specific shapes. So we were trying to actually make a molecular cylinder or a trough that would carry something. We looked at molecules that would interact with C5a, which is another G protein-coupled receptor associated with inflammation, associated with the recognition of molecules. We're looking at how we can constrain those molecules to adopt a specific shape. Again, looking at those cone snails that kill you. Further GPCR stuff. Looking at molecules around adrenaline. So the title of my talk was Looking at Molecules. And that's what I spend a lot of time doing, or at least I did. These days we get into other areas of teaching and things like that and admin, but I still find myself going back to it and looking at things. I did want to call myself a TV chemist, but a very close friend of mine coined that term so I can't use that. But looking at molecules is basically what this involves in a very collaborative fashion. This was roughly around the time that I came to Dunedin with Trudy, and probably around the time that I bought my first wetsuit, at least long wetsuit, because you didn't really need them in Brisbane. So I want to talk about some of the research that I've been doing, particularly here and still doing at the moment. What you can see here is a video that I did not make, and this represents 50 of the most abundant types of molecules in bacteria. And I'm going to stop that again because I'm going to get dizzy. And what that's supposed to show is the complexity of the cell, or a cell. As a chemist, I look at one of them at a time. Biologists and colleagues look at multiple things and pathways, and they can all work that out in their heads. And I struggle to look at one, followed by the next one, followed by the next one. I look at them separately. But specifically, just a little bit of background. We know that microbes lead to infectious diseases. There's bacteria, there's fungi, there's viruses. And they mutate, they change. And that's how we get drug resistance. I probably don't need to advocate how bad this can be, but we know it's a significant problem. And of course, the best scientists haven't been able to save their PowerPoint presentation, because I did actually add in a picture of coronavirus, but obviously it didn't save. So that's just to emphasize the fact that we're well aware of what goes on with respect to resistance and different variants, etc. So I've kind of lost track of that time. Not that time. I believe I was in Dunedin at the time, but a very good colleague of mine, Willa Houston, who was at the time in Brisbane, contacted me and said, wonder if you'd be able to help me with some work that she's been doing on chlamydia. Now, she's a very good microbiologist and was looking at the organism itself, specifically around the disease, but also because it affects koalas. Now, chlamydia, I'm sure we're aware, is a sexually transmitted disease. It's the most prevalent bacterially sexually transmitted disease. It is treatable. However, it's often asymptomatic. So that leads us to potential long-term health problems, such as pelvic inflammatory disease, infertility, sterility. So they're not good. So I thought this is going to be a decent project. And I've talked about homology and proteins looking the same. So this was all about this specific protein in orange. And it's a protease, surprisingly enough, so it recognizes something in an extended or string or line conformation. And most proteases are similar, right? So this is another protease. This is a common protease that we have in our digestive system called trypsin. And if you follow that top arrow there, it matches that. So this should be easy, right? We're just dealing with another enzyme that I've looked at before. I think I'm going to say this again, but that wasn't that easy. Because there's our classic fold or protein structure, but on the left is a blue one as well, so it's got an extra domain. Not only does it have that, those purple things activate it. It turns it on. Technically, those two purple things should be connected. So that's one big long thing which we don't know exactly how it fits, but we're aware that that's happening. It also hangs out in threes together. There's nice symmetry there. It also hangs out in sixes, in a hexamer, and it's inactivated. So we're dealing with activated and inactivated forms. How is it inactivated? So the white unit reaches across this space and blocks another active site where all the chemistry happens in the enzymes, effectively reaching out and blocking it. So what we needed to do from modelling, noting that the chlamydia enzyme is not available as a crystal structure, so we had to model these things. In order to do that, we wanted to be able to put our natural binding substrate, the peptide that this protein actually cleaves, into the binding site, but you can see that this orange bit is in the wrong place, or I can explain that it is in the wrong place because it's inactivated. So this is what we're dealing with, a protein that moves between inactivated to activated. So finally we were actually able to get something to look at as a molecule to be able to take it to the next step of designing molecules to have something that's going to hopefully affect chlamydia. It also hangs out in 20-formers. So there's 24 single units of that same protein that acts as a chaperone as well. So we had to put that aside because that was someone else's scientific research that they could look at. So this has been a very successful collaboration with Willa Houston and ongoing with close collaborators in Dunedin also. So one of the key papers was the identification of an inhibitor, which Willa identified specifically when she went to Matt Boggio's lab in Stanford. Now, I like to hope that I'm able to put people together and help people out, whereby if someone says, do you know someone who makes this type of thing? I can do that. In this case, I gave Matt a call out at a conference as I was speaking like this, and Matt had already given a talk, and I said, Matt, we'd like to talk to you about this. And sure enough, Willa went over there and identified some molecules. I believe that's actually very close to it there. So that was managed to be lethal to chlamydia, which was very positive. Now, I'm still fascinated by science. What this picture is up here is these are chlamydia cells, and chlamydia only exists within other cells. So these green dots are chlamydia cells within human cells. As I think mentioned earlier, also aspects with chlamydia infections in koalas, and they are a threatened species as a result of that. So a good friend of mine at one stage said, rather than outsourcing people to make these peptides for you, why don't you do it yourself? So we did. I went and had a chat with a good colleague, Alan Gamble, and he agreed that we could get some students to make some of these molecules. And our first one was Deji, probably the best dressed PhD student I've seen. He would walk around the corridors wearing a suit. That's him casually submitting his PhD. He did put a tie on that day, but the suits were fantastic. So he did some very good work. It's times like this where you need to acknowledge people, where only one line can acknowledge 3.5 years of their work. But he did some great work. The next PhD student who did honours, and they overlapped, was Jasmine Huang, who is currently in the States, but she had a number of papers who did some more analogues of these compounds. And when Jasmine was starting her PhD, she said, I'd like to do some formulation as well. And Alan and I looked at each other and said, sure, but we don't know really how to supervise that. So we reached out to another colleague, Dr. Arlene McDowell, who agreed to help out. And this was over COVID as well. So Jasmine was going to travel to Sydney to work with Willa to test some of her compounds and her formulations as nanoparticles to see whether or not we could get better delivery of these drug molecules into the cells. She couldn't go there, so I probably incorrectly determined that Helicobacter pylori, a bacteria that's related to stomach cancer, is also an intracellular bacteria. Putting my naivety aside, it worked. So we managed to get another paper targeting Helicobacter pylori. And I guess, for want of a better term, as funding has not been that favourable with chlamydia, we are continuing on down the anti-cancer bacterial side, looking at Helicobacter pylori with these types of molecules. So I've just thrown this up, more of an interlude between, shall we say, microbial infections. And this is probably 2 or 3 years ago, maybe more. I probably owned 4 wetsuits at this stage. It did go up to 5, but I had to get rid of one. So another very strong collaboration I've had with my colleague Brian Monk down here, who's been the principal investigator on these projects. And this is where I've been looking at the molecules again. So just a bit of background. So we're all familiar with fungal infections in our toes, toenails, etc. But they can be a significant threat and can be fatal in immunocompromised patients. So, you know, that's not a good thing. I've also mentioned that resistance to known drugs is common because these microbes, they change. The specific molecule that these things target is a cytochrome P450 enzyme. It's associated with these sterol type molecules. So this is the actual molecule, which is the fungal equivalent of cholesterol. So we know that cholesterol can be bad, but cholesterol is vital in our cells as part of the cell membrane. So interfering with the cell membrane of an organism can help to kill it. So that's why the azole antifungals are successful, except when we have resistance. So Brian's lab, and once again, so I will point out that this is probably the second time since 2000 that I was in the lab, and most scientists in the room will know that if you see someone that's a PI in the lab with a lab coat, they're not doing any science, it's just a photo opportunity. I can confirm that that is me, and my hair wasn't dyed, it's just lost its colour since then. So scientists, and particularly Brian, use Saccharomyces cerevisiae, which is commonly baker's or brewer's yeast, as a tool to look at model fungi and look at specific proteins. In this case, we're targeting fungi. So we were looking at this protein, and this is a resultant crystal structure that came out of the lab. But early models based around other species such as Mycobacterium tuberculosis, which was available, and the human enzyme, didn't paint the full picture. So I put forward that things are homologous, similar, but they're not always the same. The very first structures of this protein show that this protein showed a membrane anchor, and that's quite significant in that field. We won't go into that. But what I want to get into is what we identified with respect to the resistance mechanism. How are these bugs resistant to these drugs? So the cytochrome enzymes always involved a heme group, similar to our hemoglobin, whereby we have an iron atom and other ligands bind to that atom. Back from my inorganic chemistry days, you can see that the crystals that Brian grew were nice red colour, so that's from the iron in there. And that's because this triazole actually binds to the iron atom in the crystal structure. And what we're able to do is identify exactly how these molecules bind, but also the roles of water molecules. So we're coming back to that water thing. I like water, I like to hang around water. With these crystal structures, we're able to identify what's going on with these water molecules. And this just shows you types of networks and interactions that occur in that. So this was another picture which I'd updated, which didn't save. So this is just a representation of the type of data that we use, whereby this green area indicates there should be something there. We've added that in the experiment. And more realistically, this is the actual software that we use to show us that, whereby we would build in a drug-like molecule around here and also add in specific water molecules that are conserved highly. All this data is generally collected at the Australian Synchrotron in Melbourne, which is quite a large facility. You can see by the two gentlemen down here, a large particle accelerator. So let's get to the crux of it. So a lot of this work was done by a very talented PhD student who had very good hands in the lab, Alia Sagatova. The group has determined structures of the model drug target, so that's the baker's yeast enzyme with at least 6 known drugs putative substrate complexes, 7 agricultural or agrochemicals, so we use these chemicals against fungus, against crops, numerous structures with known clinical relevant mutations, and we'll talk about that briefly in a moment, along with known drugs and new drug leads. Also, we've determined 2 structures from fungal pathogens. So the yeast, the Saccharomyces, the Baker's and Brewer's yeast, it's a model system. We actually want to know what happens with the infectious fungi. So this probably doesn't mean much to half the people here, but this is a mutation whereby the amino acid randomly changes, but because the drugs are no longer effective against that strain then becomes more prominent and can grow. And what we were able to do is we determine the crystal structure of the native enzyme, and basically I want you to just see that red dot there. In the mutant, we don't have that red dot there. So what we determined was, with that mutation, we lost hydrogen bonding network. And the way I describe that is if you've got 3 people trying to get across a river, you hold hands, and it's much safer. It's a small river, but just work with me. If someone doesn't hold hands, you've lost that interaction. So there's not anywhere near as the strength as what it could be. So that's what happens when this particular mutation occurs. And this mutation is present in many fungal species, including the agricultural ones that we've looked at. And that's related to these short-chain azoles, such as fluconazole and voriconazole, which you may or may not have heard of. I'm sure you've used them, compared with itraconazole. So this one relies on that hydrogen bond. This one doesn't need it. So therefore, we don't get resistance in that. So just some of those papers that we've put forward. Again, Alia has done a lot of this work. There's a few more students in there as well. Brian, thank you very much for that collaboration. It's been very fruitful. Also Mikhail there. Just also to point out the homology of how similar things are in nature. So these are 3 different species of fungi. And to all intents and purposes, it looks the same. When you drill down to specific loops and amino acids, you can see differences. But it's very similar. So I'll quickly go through the next few projects, I think there's only 2. Just bear with me. So Mark Hampton, in the top right up here, contacted me. It was probably around 2008. Said, would you be interested in helping out with some modelling about these compounds? These compounds identified in cruciferous vegetables such as broccoli, cabbage, all those good vegetables that we love to eat. And they have anti-cancer and anti-inflammatory activity. So they identified the main protein target, which was macrophage migration inhibitory factor. Such a big name, that's why we call it MIF. And this was the modelling that I did in that paper. The key point here is I wanted to put forward that we also did the crystal structure, and lo and behold, and thankfully, I was right, mostly. So that was always, it's always good to know those things come out correctly in the end. We've continued on with that project with a number of, another publication with a number of molecules that we looked at. Now you remember the story with the chlamydia enzyme where it wasn't straightforward. Well, welcome to another story where it wasn't so straightforward. Probably difficult to discern the difference between the yellow and the orange portions down here. Needless to say that they bind differently in the same crystal, which is unusual, interesting from a scientific point of view, but frustrating and annoying. So that added another level of complexity of understanding this system. I have another PhD student, Lahitha, who has recently finished, and if she's in the audience or listening at home, that is next on my list to get that manuscript out. I'm also acknowledging Rob Smith and Sigurd Wilbanks, who've been very helpful in that enterprise. So I mentioned my swimming partner a while ago, Michael Kelso, who's now a preclinical trial expert in a biotech company looking at oncology drugs in Sydney. At one stage we got involved in looking at this type of molecule. So this is amiloride. This is a drug that's available on the market, and it's used for hypertension and heart failure. And the concept of drug repurposing is not unusual in the scientific field, because if you have a drug that's already been through clinical trials and you can make it work in another area, you don't have to go through clinical trials. We know that it's relatively safe in humans. So this diuretic had possible anti-cancer leads, was a possible anti-cancer lead. And with Michael, we went on to publish this article looking at a number of different analogues as inhibitors of this, remarkably, a protease. And this is the same structure as the previous ones. So they have homology. So these are probably two of the smartest people that I know, Mike Kelso and Greg Cook. And at one stage a few years ago, Greg Cook said to me, "Do you know anyone who makes any amiloride analogues?" I said, "Here's something we prepared earlier." And sure enough, after a number of years of research and testing, we've published an article targeting Mycobacterium tuberculosis. So these types of derivatives, the right-hand side of this molecule is the same as this. And there's another manuscript under review whereby they are active against tuberculosis. Again, significant resistance in this, with this bacterium, Mycobacterium, as a major issue worldwide. So I was going to put up all of the structures that I've either published, worked on, or been involved with. Technically I have. This one down here is a number of structures also, but there's 57 different protein structures that I've been involved in. I didn't put the last half of one up there, which is still on my desk, which I've got to get to in a couple of weeks, just to finish off. A lot of those are the yeast enzyme, but there's HIV, there's another different target there, a few MIF enzymes. One of the good things I saw in a lecture the other week is you can't publish these types of structures currently unless you make all of the data freely available to the scientific community. So on the protein data bank, as I alluded to before, there's over 100,000 slightly different crystal structures of potentially druggable targets available to any scientific aspect in the world. So I don't look at these a lot, but these are the types of molecules that I look at. I want to also acknowledge Dr. Andrea Vernal, who I have an ongoing collaboration with. I haven't talked about G-protein coupled receptors much, but this is one of her fields, G-protein coupled receptors, whereby I help out with the modelling and the students. In particular, not so much looking for a drug, but investigating chemical tools to better understand the disease processes and cell signalling implicated in various diseases and conditions. Specifically at the moment we're targeting the adenosine receptor. That's the one that the caffeine hits in the morning. Also the cannabinoid receptors with respect to disease and pain. Now I'm sure thousands wouldn't believe me, but I did actually add on the students here who was Smeek, China, Ian and Anna Cooper on there, who have all worked very hard on this, and they're the students that have completed their PhDs in these projects. So there's an idiom from the Maori language, he tangata, he tangata, he tangata. It is the people. Particularly for me, I collaborate with others. So these are people that I have worked with over time. A lot of them I have published with, some of them not. But up here we have, I guess, my honours. It's over there. My honours supervisor, Glenn Deacon, I'm hoping. My first mentor, David Fairley. I won't go through all of them. Don't panic. There's a couple there that I may have forgotten, but please know that you're there. Wait, there's a second page. So these are, again, people that I've worked with, the students, and people that have helped me as well. As is always the case with a scientific presentation, one needs to formally acknowledge the researchers that you work with. So these are the people that I've worked with at UQ, Willa at UTS, Monash people, here at Otago, Edinburgh, Ottawa, Victoria. And just as a quick note, the superscript for most of those means that they're no longer at that institution. And of course I missed a couple as well. Also need to recognise the funding bodies there that help support the research and the collaborators that put in those grants as well. So let's go back to a bit of the water. This is what I do when I'm not here working. I don't need to convince most people that when you wake up on a Saturday morning and you get to look at that, that's pretty cool. Particularly when you've got people like this doing stupid things out in stupid surf. That's at the National Championships. Sorry, I'm just losing my voice. This is what it looks like after 2 hours of paddling fairly hard in a waka, which was 2.5 years ago in the Marlborough Sounds. Most of the time we prefer to have water like this, which is nice and flat. So we managed to do a paddle from Cromwell to Clyde. These are before and after shots. So this is me in the fourth, was that the fifth wetsuit? I'm not sure. This is the after shot. That's all right. Thankfully, the runner is with us today after looking like death, having just run a half marathon off-road. We're also going to do our 11th triathlon in a month or two. And that's a significant collaboration that we've had continually for 10 years. Last one. So these are the people that keep me going. So thank you very much for doing that. This one here, I'll just point out that it was too cold to go outside in the snow, so we thought we'd toboggan inside. Also, my daughter taking one for the cause. The prize was beer for her winning a race. So we took that on board. Special thanks to these people, Trudy, Lara and Sam. As I said, you keep me going. Mum, Dad and Di, give you a wave. Dean and Kelly, thank you for coming. Val and Gary, great to have you here. Latta and Milne crew, also great. As I said, collaboration of 10 years strong, if not a little worse for wear, with the triathlon team. A few good knees. Little did we know that I would also get a broken one too. All the rest of the people up there, and these are some of my happy places. So this is Glendhu Bay. That's actually just over there in the harbour on a fantastic day in the middle of winter. And thank you very much for listening.
Good evening, everyone. My name is Brian Monk. I'm a member of the Department of Oral Sciences, and I'm in the Sir John Walsh Research Institute in the Faculty of Dentistry. And Joel is a great collaborator. He collaborates with almost everyone. I'm surprised if he walks down the street, he doesn't start collaborating with someone else. Joel's arrival in Dunedin with his wife Trudy Milne in 2014 has proved to be an excellent career move. In addition to raising their family, Trudy has spent almost 2 decades carrying out research at dental school. So that was a good one. And she's done that on a wide range of projects. In the meantime, Joel established his academic credentials in the School of Pharmacy across campus, nationally and internationally, and he'd put a fantastic start into that even before he got here. In addition to his teaching and administrative responsibilities, Joel has successfully assembled a computer-based facility that has enabled him, his colleagues and students to visualize and understand and use effectively structures of target molecules and their ligands, and understanding the chemistry of what goes on in those connected structures. As my group became increasingly interested in using such valuable products of structural biology in order to complement the sort of stuff we do, which is to look at phenotypes and how we can throw drugs at them and make things really sick. Or, for example, trying to understand drug resistance, as Joel talked about in his talk. It was really great having Joel, the structural biologist with a medicinal chemistry background, just across the other side of Frederick Street. It proved to be very, very convenient in many ways, and it's certainly a win-win for my group and the several students who we co-supervised. So they had some great supervision. Joel's lecture this evening has clearly illustrated and communicated just a few aspects, I think, of the high-level contribution he has made to several research groups and across a broad spectrum of problems and projects. It's been a fascinating opportunity to listen to you cover that broad spectrum, but you haven't done it in particular depth, so I look forward to hearing that in the future. In some cases, of course, Joel's structural studies, his model building and homology model building, and docking studies have been of immediate value. You can go out and you can find molecules that might work. And that's really great. But even if the answers are not immediate, of course, there's enormous value. The information created often helps frame new and more insightful strategies and questions, and he's been doing that throughout his career, and it's certainly helped with the grants that we've written locally in terms of having that structural background there and having his support. Finally, doing the mahi or the work by creating libraries of target and target ligand structures and models and publishing such information and placing that information in curated databases, such as the Protein Data Bank, contributes to a pool of accessible biological information that will drive future innovations and solutions, such as novel drugs, as well as providing the basic knowledge needed by evolving technologies such as artificial intelligence. We've got to have that basic knowledge out there, and Joel has made a great contribution to that, which I think he should be very proud of. The adage that a picture is worth 1,000 words is clearly at this stage an understatement. I'd like to conclude by just talking a little bit about where Joel came from. He's a graduate of Monash University. He graduated there in, I think, 1994, before going on to a PhD in Queensland. So Joel completed his BSc Honours at Monash 17 years after I completed my PhD there. And I just want to talk about an incident that briefly illustrates how we work on scientific problem solving and just how small the world we are in actually is, because Joel collaborates with everyone, so obviously it doesn't take 6 people in the world that you know to get to Joel. It takes about 2, so I hear. So the story is that about 3 weeks in about 1974, the Fire Brigade attended the Monash University Biochemistry Department where I was doing my PhD. And they attended that building 3 times over 3 weeks. In each case, it seemed to be a false alarm. On the 3rd Friday afternoon, visited by the Fire Brigade, the culprit causing the call-outs quietly identified himself to his fellow PhD students. I don't think he identified himself to the teaching staff. And he also put in place a 100% effective solution to the problem. How did he do that? What had happened was that every Friday, getting towards the weekend, he needed to disinfect his bench. And so what he would do was he'd put some ethanol out on the bench, and for some unwarranted reason, he would get out his Bunsen burner and flame it. And what he then realised was, after 3 weeks, that he was making this little conflagration that went up, and there was a nice little heat sensor up there that set off a fire alarm at the fire department. Importantly, the student culprit had successfully interpreted a serendipitous experiment which was replicated 3 times, and What's more, had implemented a suitable course of action. That incident clearly shows to me that Monash University to be a great learning environment from which Joel benefited in the 1990s, taking him from economics into science, a place where science is done every day in an everyday manner. And of course, the place provides all the instruments you need, including a fire engine. But anyway, furthermore, the molecular models and images that Joel prepares and shares so frequently illuminate the way forward. And they usually glean from hard-won and sometimes serendipitous structural information, often obtained at the Australian synchrotron located adjacent to Monash University. So that university still has a great impact on his life. But I know that Joel has a great impact on Otago University, and I'm very proud of seeing what you have done. By the way, I should go back to the student culprit, which is another link. The student culprit subsequently had a stellar and, I believe, fire-free scientific career, including helping establish and then heading the prestigious Institute of Molecular Biosciences in Brisbane, where Joel and Trudy were employed briefly just before coming to Dunedin. So you know who that is. Okay. My final task is to acknowledge and thank Joel for his excellent and informative contribution this evening, and to present him with a token of our appreciation, which I understand has been stashed down here. So, congratulations, Professor Tyndall.
All right, so, there we are. Tēnā katoa, ko Neil Gemmell tōku ingoa. So I'm Neil Gemmell, I'm the Acting Deputy Pro-Vice-Chancellor of Health Sciences, and it's my great pleasure to bring things to a close by first thanking Joel for a wonderful lecture, and also thanking my colleagues for their remarks opening and closing the event. I am stunned by the work that Joel has presented looking at molecular interactions, and I think most importantly the interactions among people, which is mirrored in the work that he does. And so I think, just to conclude, I just want to say please continue the interactions with Joel and whānau in our staff club, where there will be food and beverages available. Kia ora.